Hello, and thank you for joining us today for this Dovetail Genomics webinar hosted by Samba Scientific. My name is Jason Amsbaugh, and I'll be the moderator today. In this webinar, Dr. Giacomo Cavalli, the head of the Chromatin and Cell Biology Lab at the Institute of Human Genetics, CNRS, and University of Montpellier, will present recent studies using high-C technology that reveal specific roles of structures such as topologically associating domains, or TADs, and chromatin loops in gene regulation.
So thank you very much for the invitation, Jason. It's a great pleasure to be with you today. I'll start sharing my screen right away. Let me know if you see it.
Looks good.
Okay, so yeah, I'm gonna talk about chromosome organization and some specific epigenetic regulators of gene expression, in particular called polycomb proteins. So I'll give a brief overview on these proteins, and then we'll dive right into science. So what we've learned in the last sort of maybe 15 years or so, thanks to these new technologies like high resolution spatial technologies, is that the genome is organized in a hierarchy of structures. And if you look at the relatively short range, here we are seeing a region of about half a megabase. You see these specific contacts of high frequency contacts. So this is a map of high C. So you see the coordinates, you see some tracks of epigenomic, interesting epigenomic components like CTCF and histone marks. And then when you have this red signal, it's a strong signal. For many of you, you will be probably acquainted to these kind of maps. And these interactions, we call them loop because there's a strong interaction flanked by nothing much, so the rest of the point. So the easiest way to represent is that you have a contact like this. And this can be happening at CTCF binding sites. So CTCF is a main organizer of chromatin folding, assisted by cohesin, enhancer promoter interactions, or polycomb-mediated interactions. And I'll be talking about these today. And if we zoom out now this region here becomes a tiny region in an 8 megabase region of the genome. Now what we see is a new feature, which is the fact that you have some domains where you have a relatively high frequency of interaction everywhere within the domain. And as soon as we move out of the domain and we look into the flanking paths, we have some other, these sort of, let's call them triangles, that have high frequency interactions within, but interact much less with the neighboring domains. So these have been dubbed in multiple ways: physical domains, chromatin domains, and maybe the one that is most used is topologically associating domains, or TADs. If we zoom out even more, we see that there's yet another structure. So now again, this region is part of a 30 megabase region that is larger. And what you see is that individual TADs or flanking TADs form what we call compartments. And basically what many people have found is that there are regions where you have high levels of open chromatin marks that interact with other regions that also have the same open chromatin marks, while the inactive regions that are devoid of marks or are maybe coated by K9 trimethylation like to interact with other regions of the same kind. And so what has been used as a nomenclature is A for active and then B for inactive compartment. But you will learn that there's even more than this, in fact. So among the regulators of all these architectures, I told you of CTCF, and polycomb proteins are also an important class of pleiotropic regulators. They regulate many genes. They were discovered initially in Drosophila as repressors of important genes, the Hox genes, which determine the anteroposterior body plan of the embryo and throughout development up until the adult. But then later on, when high throughput techniques came out and people could start looking first by chip-on-chip and then chip-seq, we and others found that these genes are bound to many other regulatory genes like cell cycle genes, transcription factors, signaling genes, and in that way they regulate many processes in the organism. including proliferation, cell fate, many developmental pathways, stem cell plasticity and differentiation. And when this is regulated, there's several diseases like cancer that come up. So one important point to keep in mind for these proteins is that once they start to bind to a gene, they have the capacity to maintain their pattern of binding and the silent state of the target gene. So for instance, if you look at the initial development of the fly embryo, but also of mammalian embryos, you will have polycomb proteins everywhere, but where the genes, the target genes, for instance, Hox genes, have to stay repressed, they will bind to the repressed targets and then stay bound throughout the cell lineage until the end of development. And in that way, they maintain the memory of the repressed state until adulthood. And there is an opposing or complementary class of proteins called tritox group or compass proteins, which have chromatin remodeling and histone modification, like histone methylation and acetylation abilities that are doing the opposite. They're maintaining the on state of the same genes in the right place. So how do polycomb proteins act? There are multiple ways in which they, first of all, find their targets and then repress their target chromatin. So, go back. So the targeting, they can either use transcription factors to be targeted, or they bind to simply to unmethylated, so non-DNA methylated CpG islands, so CG-rich DNA. So this will attract initially a subset of the polycomb proteins that are forming a complex called non-canonical PRC1. And this complex is depositing a histone mark, which is H2A lysine 119 ubiquitinated, which is recognized by a subcomplex of the PRC2 family. And now this one is going to deposit another mark, the K27 trimethylation. And K27 trimethylation in turn is recognized by itself and by canonical, or the first actually variant of the polycomb class, is the canonical PRC1. So this one then has forms a sort of self-enforcing loop. So once you start recruiting polycomb proteins, you start with a variant PRC1, then you get PRC2, then you get canonical PRC1, and this all reinforces the binding of the others. So in this way, you can induce chromatin compaction in cis in a relatively short range, but you can also counteract activating, so opposing activating complexes like SWI/SNF or CBP. So you can have a direct competition mechanism to repress their target genes, but also these proteins are forming hubs in the nucleus, and you will see some of this. And in this way, they can also exclude activating components. So there are multiple ways in which they can act to repress their target genes. And you will see there's some surprising non-repressing function as well of these components. Now, if you look in mammals, I've been in a way simplifying all this plethora of complexes that is formed by PRC1 components and PRC2 components. Indeed, they have multiple variant complexes in both cases. And what makes things even more difficult is that for an individual subunit of a complex, this is for instance the canonical complex, but in fact, this PCGF, in fact, there can be 6 different paralogs forming it. And you can imagine all possible combinations of them, which makes the combinatorics quite difficult to study. The flies are a little bit of a more tractable system to study polycomb function. They have all the conserved components. They also have variant complexes, but the advantage is that in most cases, for most subunits, you only have 1 or maybe 2 genes coding for it. So it's easier to do the genetics. And that's why we did quite a bit of work in the fly system. And I'll start looking into loops that involve polycomb components. And this is a work that has been done in the lab by several people collaborating. It was a very nice internal collaboration. So as you see here, if we take a fly embryo and look through development, and fly embryos develop super fast. So you can, a 2 hours old embryo already has over 1,000 cells, about 1,000 cells. What you see is that you start seeing loops, and if you look at the screenshots of chromatin marks, you see that many of these will have K27 trimethylation. And so this is another example here, and this is a locus that we studied in more depth. And once the loop is formed, it is maintained throughout embryonic development. Actually, even these late embryos, they're still 1 day old, and they have about 50,000 cells. So what can we do to perturb the formation of these loops? Well, what we did was to insert between two binding sites that are spaced here about 60 kb, a barrier, a strong insulator that binds a DNA binding protein called suppressor very wing. It's in a way a sort of fly homolog. It's not a direct homolog, but it works in a similar way to CTCF. So once we insert this, the first thing to check is whether anything happens to polycomb. So this is just the Q chip showing that with or without the insertion of the insulator, there's no change either in polycomb component binding or in K27 trimethylation. But because of the binding of this insulator protein, if you do a microscopy, this is fluorescent in situ hybridization against DNA probes with the two sites that are bound by polycomb. Normally they are highly colocalized in the nucleus, but if you insert your insulator, this colocalization is reduced or lost. So, and we see the same also when we do 4C-seq, where we see that there's a decreased interaction between the two. So what happens now in terms of function of the underlying genes? And actually there's a gene here, which is in this region of the locus, which is an important gene for leg morphogenesis. And in particular, this gene is normally expressed in the proximal part of the leg, and it's important for the formation of this little structure here, which is, however, an important sexual trait of Drosophila maize. And if the gene is overexpressed, you see extra traits in the distal part of the leg, which you shouldn't normally have. And when we insert this insulator, what we see is that in a fraction of the flies, you start to see this structure, which you shouldn't see. However, as you see, this is only a minority of the flies. It's less than 10%. And what this is telling us is that when you perturb the 3D architecture of these complexes, you still maintain gene expression or gene repression to a good extent. However, you are reducing the fidelity with which silencing is maintained. And so in this sense, you can say that although there's a lot of regulation in cis, polycomb-mediated loops can play an instructive role in gene silencing. So, but we got actually interested in another function of these proteins as well, because when you make mutants of polycomb, you typically have homeotic transformations. They are very severe. Null mutants die as early embryos, but you can also make tissue-specific and stage-specific mutations. And in this case, you start to see other phenotypes. And one very striking phenotype that you see if you make mutants specifically in a tissue like the eye precursor tissue, which is here. These are larvae which are normal, so we can induce a clone of neutral cells that are not mutated. They are just marked by GFP and they get normalized. But if instead you ablate PRC1 by null mutation in a critical subunit like a polymeiotic, now you see that these clones are growing huge. So they are marked by GFP and they are really invading the whole tissue. And actually most larvae are killed and only 10% survive, but they have severely abnormalized. We induce the mutation only in the head of the larva in the eye precursor tissue, but if you combine this with an activated oncogene, you will see that you get metastasis all over the body of the larva in the posterior part here. And we also saw that these phenotypes are dependent on multiple signaling pathways, like the Notch pathway shown here. When you reduce the Notch signaling, you also restore the normal eye or almost normal eye. And the same is true for the JAK-STAT pathway. So This is true for this particular subunit, but also for other PRC1 subunits. So again, normal tissue, and these are these abnormal large tissues with PRC1 subunit mutations. And it is not, a fly oddity, because if you take cancer cell atlas data, and then you look into multiple cancer here, like for instance, acute myeloid leukemia or bladder carcinoma or breast carcinoma, and you look at the level of expression of multiple PRC1 components. I told you there's a large number of paralogs, so we have to look at all of them. You see that many of them are downregulated, and if you, this is the compiled tumor data for the one of the subunits, PHC1, sorry, and you see that there's globally a downregulation in the tumor sample, and the same is true for breast carcinoma. And if you now stratify the patients, those that have the lowest expression levels of this subunit have also the poorest survival. So this really suggests that you might have a tumor suppressing function for these subunits. And the same is true for many others that we've looked. So this kind of tumor suppressive function is something that is conserved in some of the human tumors. Now, of course, it's very important for us to understand how this works. And so we did a lot of genomics, and we mapped in detail the binding mode of these polycomb components, but also of histone marks in this particular tissue. And what we saw is the expected binding in many genes. So this is a clustering of the binding modes using the histone marks as classifiers. And as you see, is that you have a lot of polycomb components in regions that have high levels of K27 trimethylation and the H2A ubiquitination as expected. But you also have many unexpected binding sites. Some of them are these, which are enhancers. There's basically places where you have PRC1, low levels of PRC2, but you don't have K27 trimethylation, low levels of ubiquitination of H2A. And instead, what you have is a lot of active marks like K27 acetylation and K4 monomethylation, which are really the enhancer distinctive marks, and you don't have elongation or promoter marks in these places. And they actually correspond to previously mapped enhancers. And then you have another category here, which are actually active promoters, which are marked by the promoter marks. There is elongation close by because it's TSSs, and there's very low levels of K4 monomethylation. So, arguing against the fact that it would be enhancers. So what is Polycomb doing at enhancers and active promoters, which was not expected? So we did IC, and you probably, some of you might be acquainted now with this association by contact kind of paradigm. So the idea is to see whether you have association of given enhancers with promoters, looking at the levels of contact, and that's what we did. But we stratified the enhancers and promoters, and we were looking by the amount of polycomb bound to them. And as you see, in particular for the enhancers, the more you have polycomb bound, the more they contact the cognate promoter. So, polycomb is really accompanying enhancer promoter interactions in active genes. And what are these active genes? If you do gene ontology, there's a lot of the ontologies are actually eye development, eye morphogenesis, or cell fate. So it is really important genes for eye differentiation. And again, what is polycomb doing? So what we had to do at this point is make a mutant and look in the mutant how do we change expression of these genes. And as I told you, polycomb is normally a silencer. So if you do this, you will induce the expression of Hox genes and many other canonical targets where you have K27 trimethylation. But you don't do that in these places where you have enhancers, developmental enhancers, and promoters bound, and instead these genes are going down. So what this is all suggesting to us is that, yes, you have repression, repressing hubs, but you also have active enhancer promoter contacts that are mediated by polycomb proteins. So you, at the same time, you maintain cellular memory by maintaining silent chromatin states, but you can also accompany activation of target genes in a subset of the targets which are developmentally important and prevent, by the fact that they drive differentiation of cells, they prevent cancer. And actually this leads us to a final point for this part of the talk, which is, but do we actually need mutations? Because as you've seen, with a single hit, you get a full-blown cancer, which is something that is a bit unusual for at least for people who study human cancer. Typically, what we think is that cancers are a genetic disease, and a single hit is usually insufficient to induce a cancer. You expect that you have multiple hits before you can obtain a cancer. On the other hand, now if we look at this is the famous drawing picturing epigenetics, where people think, okay, we have your zygote here, which is going down the developmental path, and it finds different bifurcations, and it can take different fates. That is going to be the differentiated cell types. And epigenetics is the study of everything that can modify gene activities in the context of the same DNA sequence, right? But then we can also imagine as a corollary that if we force cells to take an altered state, so let's suppose they should go this path, but now we push them to go another path, even if it's a transient phenomenon that pushes them, once they are down the line, even if you restore the initial condition, will they be able to go back? And as we know, when we do cellular programming, going back is very hard. It's not impossible, but it's pretty hard. So what happens in a situation that we would modify like this? So the first thing that you have to do for this is, what about, instead of having a null mutation, we just make a downregulation of our components? And as you see here, this is again a control tissue. A simple downregulation is sufficient to induce a full-blown tumor. And so now what we did was to develop an in vivo system where, instead of completely depleting a component all the time, like I just shown you, we can deplete it, but for a window of time of choice. And it's a sort of, if you wish, for the people working in mammals, like an oxygen system, but you can use it in vivo. And it's a complicated genetic makeup. For the fly aficionados, it's here. It's using an exogenous transcription factor GAL4 that is inhibited by the GAL80 inhibitor, but this GAL80 is temperature sensitive. So what we have is at 18 degrees, there's no inhibition, but we can move the organism anytime we want at 29 degrees, keep it for, the minimum is probably 16 to 24 hours, sorry. And this is gonna repress the expression of the protein of choice. In our case, it's a polycomb component. And then you bring the system back to the low temperature, and your protein is restored. And flies are happy at both temperatures. We did all the controls, nothing happens. So what do we get? So this is again a normal tissue. These high precursor tissues are epithelial cells, and it's actually a bilayer of cell in this initial stage of development that has a very nice apical basal structure, which is marked by actin. So this is what you see in the normal tissue. If we induce a tumor by constant depletion, cells start to stack on top of each other. They are no longer contact inhibited. They lose the polarity completely, and so this is generating these huge tissues. What happens if we do the transient depletion for only one day and then bring the system back? And strikingly, what we get is, again, a complete loss of organization of the tissue, with all the cells that are the loss completely the orientation, hyperproliferating. And actually these tissues, they grow large. So this is the constant depletion where the tissue is growing big. Just to give you a scale for flies, this is happening in 1 week. Okay, so it's even less than 1 week is sufficient. And if you do this transient depletion, this is for 6 days, this is for 8 days. Actually, the more you wait, the more the tissue grows, and it will finally kill the larva. So you cannot even study that further than that. So basically, this is telling you that cell differentiation can be perturbed in an epigenetically heritable manner to produce cancers in the absence of a mutation, just by manipulating the levels of expression. And so to summarize this part, this would be your Dovetail landscape. Let's suppose that this region here is the normal cell fate region, but you don't want to go to this part, which would be like the disease part. But now, what happens if you modify polycomb, if you deplete polycomb? Well, what you're going to do is to modify the landscape. And so now this ball starts to roll the wrong side. And once it's here, if you maintain depletion stably, there's no other choice but just keep going down and you get a cancer fate. But even if you reestablish the polycomb system, if you do it too late and the cell has already traveled, yeah, you get a reestablishment of the landscape, but the cell still cannot jump the way back and will still go to a cancer state. So the time is a very important issue. If cells already differentiated too far, even reestablishing the components is not enough. Okay, let's go back to the mammalian system and dive a little bit in the mammalian levels of genome organization and gene regulation. And let me actually focus a second on the TADs. TADs have been shown to be important regulatory landscape for gene regulation. And this is an example from the Mundos lab, which is a classical, it's become very fast, a kind of textbook example. These are two TADs that are flanking each other. One is the PAX3 TAD, and we are looking in the limb bud, and PAX3 shouldn't be expressed in the limb bud. So it's, as you see here, very weak, just a little bit in the proximal part. And then you have a neighboring TAD here with the EPHA4 gene that has its own enhancer. This guy is expressed in the limb bud, and the enhancer doesn't reach PAX3. But if you delete a portion that encompasses the whole EPHA4 gene and the boundary, now, and you perform, this is now a 4C experiment, you see that you start to have contacts between the PAX3 gene and the enhancer of EPHA4. So here, basically you have a hijacking of the enhancer, and this is activating PAX3, and you have a digit malformation. Now, this is only happening if you include the boundary in the deletion, because if you have a large intra-TAD deletion doesn't include the boundary, you don't change the contacts with the enhancer, and your limbs are almost perfectly normal. So this is suggesting that these boundaries are important for gene regulation, and people have found it in many systems. But all these studies, they are done using very large population of cells, millions of cells. Our TADs actually folded in single cells. And so we did that by looking at using imaging, and in particular super resolution imaging, different kinds of. We used structured illumination microscopy, which I'm going to show you now. But we did the same using STED, stimulated transmission depletion microscopy, and we got very similar results. And what we see is indeed TADs are sort of separated in nuclei. So if you take, sorry, the pointer is a bit too sensitive. If you take two probes that are in the same TAD, because they're in the same TAD, you expect them to sort of intermingle if you do a fish microscopy and look by super resolution. And that's what we see, in fact. So this is a TAD of about half a megabase. And if you overlay the signals in green and magenta, you see a lot of white. That's the level of overlap in multiple nuclei. And these are actually segmentations of multiple nuclei, individual nuclei, just to show you that, yeah, you have variability in different nuclei, but you have a lot of interdigitation between probes that are in the same TAD. However, if you take probes that are in neighboring TADs, you see them very nicely separated, right? So these are actually averaged signals, just to give you an idea how much overlap there is. And you see very nicely that there is separation when you take different TADs. So TADs are sort of nano compartments, if you want, in the nucleus, but there is also variability cell by cell. There is something else which is striking to the eye, however, is when you take a single probe that is spanning the whole TAD, you might have expected, well, maybe it's going to form a globule, right? There's nothing special in terms of contact. There's no, in particular in this stud, there's basically no CTCF. There's very little histone marking at all. There is no gene expression. So we could have a globule, but in fact, it's not the case. What you see here is very variable folding. And you see sub-objects that you can actually identify and segment very nicely. So you can, this is watershed segmentation. You can identify individual compartments, sub-compartments within these studs. So is it something that that can be changed, that is regulatable? And yes, we found at least one thing that changes the number of subcompartments, which is the levels of histone acetylation. When we expose these cells to a TSA, what we see is very clearly, there's just one example, that your TAT probe is really opening up and having way more of these subcompartments. That you can do that in looking at multiple TATs, and we constantly see this kind of phenotype having more compartments, more nanocompartments. And if you think of how histones contact each other, in fact, histones can form higher order interactions by simply using histone-histone interaction. The histone H2A has a binding pocket with the histone H4 of neighboring nucleosomes. But we know that if we acetylate histones, and in particular histone H4, we are going to perturb this interaction between different nucleosomes. And so what we think is happening is that because of this perturbation of high order nucleosome interactions, you're opening up your chromatin and having more of these compartments. Nice, but do we see this only using super resolution? And so what about having another super resolution technique, but now a genome-wide contact technique? And that's where we went to micro-C. And what I'm going to show is using this Dovetail kit. We actually did a good number of optimization, and now we have a very robust protocol that we are happy to share. So what do we see when we have this ultra deep micro-C maps? And actually, people have published that before. If you look at low resolution and you compare it to the best possible high C, at least in mouse, which is this paper from our lab early on, we see kind of the similar structure. Okay, so until you look at maybe 4 kb or 2 kb resolution, there's not much of it. Maybe a little bit of a CRISPR structure, but kind of a similar thing. So here, just to give you an idea, here we have a very deep map with 22 billion reads, 6 replicates, and if you look at the intrachromosomal contacts, that's the number. But if you look at higher resolution, you immediately see that things that you are no longer able to resolve when you look at 400 base per resolution, you earn the ability to study them. And this is the SOX2 locus. This is actually the coding part of the gene. This is the SOX2 superenhancer, which is forming a very nice contact with the SOX2 promoter. And this is a region of about 100 kb, 130 kb. You see this beautiful loop. So that's very nice. Just to give you another example, that's the KLF locus. Again, at 1 kb, sort of you see there's already some difference. But what you also see very nicely is that the structures that you see here, they really start to fragment into subdomains. Of course, you can kind of guess that you have these domains, but very hard to separate them. And here it's very clear that you're forming smaller domains that are kind of chromatin nanodomains of 100 kb or less in size. And this you see in many TATs, including in silencing domains in some instances. That's another example here with the OXD cluster, which is beautiful because it has been studied in great detail by the lab of Denis Duboul. Again, that's just a comparison at low resolution between IC and micro-C. It's kind of similar. And just to show you, this is a capture IC. It's probably the best capture IC that at least I know of that people have done. It's out of a paper here that's soon to come out, but you can already find it in bioRxiv. Again, very similar kind of architecture. But if you look at high resolution, even the capture IC is at a big loss. Okay, so compared to what we see with micro-C, where you see a lot of subcontacts that you absolutely do not detect with the best IC possible, and even with capture-C you can't. But you see them now. And what are these multiple contacts? That's very interesting, because now we can overlay all our epigenomics. And so this is a gene locus which is repressed. So in DZS says there's no gene expression. Nevertheless, you already see preset contacts. Some of them are involving CTCF sites, which are bound. I mean, this locus is bound by CTCF in multiple positions. But there's more than that. This is a YY1 site, for instance, that is forming a contact where there's no CTCF with the other extreme, the CTCF bound one. And there's some of the contacts that are made, which are actually high levels of signal of ring 1b, which is a polycomb protein, instead of the CTCF, which is for some of the contacts not forming loops. Okay, so we have a mixture between CTCF loops, YY1 hybrid loops, and polycomb loops in the same locus, which is now at a repressed state. So Denis has studied the opening of the locus in great detail. I won't go into that, but you can kind of see already the complexity that you can detect. Now, the nice thing, because we have this super high resolution, is you can really delve into these loops. And just to give you an idea, we can treat again these cells with TSA. I showed you before the effect when you do microscopy. You have a big change in gene expression. Many genes are up and down regulated. But what happens in terms of genome folding? And what you see here is that you have several things that you observe. First is you kind of have an increased, the signal is more crispy. You have clearer loops. And also genomic interactions become a little bit more segmented. You see kind of here, all the signal is sort of homogeneous within these large studs, but now you see more structure, more loops, more interactions. And this is something that we see kind of genome-wide. That's another example. So, several cases you have like a trace, a shade of loops, internal loops, but they come really pronounced when you treat them with TSA. So segmenting interactions and new formation of loops. And now the great thing, I would say, with having this high resolution is that you can start calling loops. So there's a tool that people use that is called Mustache, but the tool to call loops in micro-C or high-C is usually not performing incredibly well, but it starts to do that if you use high number of replicates and either high resolution, but you can also do region capture. If you don't want to spend all the money and sequence that much, you can still do region capture micro-C and run this tool very nicely. And so here we were able to call about 30,000 loops that are really robust. They are found in all the 6 replicates. And we see that there's about 1,000 that get stronger and others that get weaker upon TSA treatment. And that's just to give you an example. This is one of the loops that is really, popping up. It's almost not present here. And we can really quantify these things. We can pile up all the loops. So we can take the loops that are expected to become stronger, and we see they get stronger, and then the loops here that are reduced or lost, and you see that there's a clear difference in the pile up. Now, there's a very interesting specific behavior of these loops, because it's not all the same. We can look at different distances, and actually, as you see here, there's a striking behavior of these TSA-treated chromosomes. Basically, they are, sort of distending. They are opening up, and so you have a reduced amount of long-range interactions in the bulk along all the chromosomes. So you lose long-range interactions, but which one? Okay, and what about our loops? And so we can look at specific kinds of long-range loops, and what we know is that there's super enhancers that form long-range distance loops, and polycomb proteins that do the same. When we look at the super enhancers, strikingly, if you treat with TSA, you increase acetylation. And so, intuitively you could say, well, supraenhancer should do fine. And in fact, what we see is that the supraenhancer contacts that are nicely present in the control cells are basically lost or highly reduced when you induce hyperacetylation, as if in fact you will interfere with supraenhancer contacts. And instead, what you don't lose at all is the polycomb contacts. So although you have hyperacetylation in bulk, polycomb is still managing to keep the silent state and make very strong long-range interactions. So this comes to the end. I think we are learning, we're still in the process of learning many things about nuclear organization, chromosome organization, and the effects it has on gene regulation. So this was like a textbook view that we can still find in some of the textbooks, in fact. We have sort of beads on a string, and then you have a 30 nanometer fiber, and then something we don't know, but some sort of loops, and then chromosome territories. Sorry, but now what we know is that instead, first of all, you don't really have a 30 nanometer fiber. Instead, you have a disorder fiber with maybe clutches of nucleosomes that get together in small groups. And then these are forming these chromatin nanodomains, which are like in the order of the gene size, or 100 kb-ish, depending on where you are in the genome. And these then together, multiple of them form topologically associating domains. In turn, then they form compartments that are either active or repressive compartments. So where are the specific contacts or loops? Well, actually everywhere. You have a lot of them that are formed between cohesin CTCF binding sites, but also between enhancer and promoter binding sites, within TADs or within chromatin nanodomains. Some of them, we actually find them at very short distances, like sub 10 kb. And in other cases, you also have ultra long range contacts. And I told you about the supraenhancers and the polycomb as an example. So, I think there's a lot to do to understand the function of all of these, but it's going to be nice work for the future. And this is just to show the lab. Very nice collaborations with Benoit Bruno, Elfej Nora, and also modeling people and imaging people in the Montpellier area. And thank you very much for listening.
Excellent. Thank you for that presentation, Dr. Cavalli. That was a great talk. All right. Thank you for that. Okay, we're going to go ahead and move to Q&A now. The first question is, how are TADs and AB compartments related or different to each other?
Yeah, so that's a great one. So they are related globally. What you see is that TADs, when they tend to have more active genes, more open chromatin content, but also polycomb actually. Polycomb is not forming super repressed TADs. It's repressing genes, but it's still in the A compartment. So all these tend to be in the A. So polycomb is in between, sort of a third subcompartment between A and B. And then the B is really regions that are gene poor, inactive, usually K9 trimethylated, DNA methylated as well. And actually, there's some nice studies showing that lines are associated with the B compartment. So probably that's actually something that I've been not approaching too much, but repetitive elements are very important for compartmentalization of the genome, and particularly for the formation of the B compartment.
Thank you. The next question, are the loops inferred from co-expression of various genes along the genetic strand represented by the line? I guess this was from one of your earlier slides.
Yeah, not necessarily, but it's frequently the case. I mean, you frequently have genes that are co-expressed and that are looping. Actually, the loop that I showed you, for instance, the case of the polycomb, I told you about one gene which has an easy phenotype that we can score. but there was a second gene in the domain. That guy, you can see the RNA expression, but you don't have very strong phenotypes when you change the levels of expression. But we've seen it's co-expressed, it's expressed in the same cells, and it's also co-regulated to a very good extent. So I would say frequently yes, but not necessarily always. I mean, or at least it's not always a perfect co-regulation. So, and there is something that we still have to learn. You may have a loop, but actually you can have a loop that turns from a repressive to an inducing loop, and so it might be that you can have co-regulation in some cells and independent regulation in others.
Gotcha. Thank you. A sort of a related question: are these looping phenomena related to zinc finger proteins or zinc finger protein loops as well?
Yeah. So yeah, actually, so because now, so let me tell you these loops that we've been calling. The nice thing about micro-C is that when you call for loop contacts, with the old days, even like our high C where we had billions of reads, the resolution was low. You had to call them with maybe 10 kb resolution, sometimes 5 kb, but that was rare. Now we can go to way lower resolution, in particular when the loops are not very distant, so we have a high number of reads. We can use easily 1 kb resolution, and so we can look at binding sites, and so we can use the epigenomics. We can see the tracks, and but also we can even look at sequences. And so some of the loops are, of course, the CTCF loops, but we can also see zinc finger protein loops. In particular, we've seen several of them that are YY1 dependent, and yeah, so that's and actually, they are partly in different, so they partly overlap, but partly they are really independent groups of loops. So that's, of course, now the big future, right? Because we can understand really the grammar of these interactions.
Thank you. So I guess there's a couple questions here. We'll take a couple different categories of questions. The first is the role of carcinogens validated by epigenetic cancers rather than cancers caused by mutations? And then some follow-ups there. What would be the proportion, or do we know the proportion of epigenetic cancers? I guess we'll just go ahead and maybe leave that question for now.
Yeah. So, of course, this is very fascinating, but very unexplored as well. What we set up is a system which is kind of a toy model, but showing really the point. And however, even we have not, completely solved it, because while we know that the induction is epigenetic, and we've actually sequenced, we took these tumors really early and done genome sequencing, and we did not find any driver, oncogene mutations in these. It could still be that later they start to accumulate mutations. So mutations could contribute even in these cases. Now, that's the case of fly. It's a kind of a toy case. What's very interesting is to look into mammals, and there's little work done. But there are some cancers like ependymoma, for instance. The PFA kind of ependymoma has essentially no driver mutations known, and there is severe decrease of K27 trimethylation, so it's a polycomb 1. There's also an example with metastases where people have looked at breast cancers, and the Weinberg lab, and they inactivated PRC2, but only transiently. And in that case, they induce increased metastasis in mouse system. But so it's human cells that are put in mouse, and they see very increased levels of metastasis with a transient inactivation. So I think it's a beginning, and one has to delve into that, definitely.
Maybe a follow-up question to that. Can we reprogram the cancer state back to normal state by changing genome architecture?
Yeah, that would be, of course, a super interesting thing. So that's something that we think at least we will be able to approach using the toy model that we have, because we have done the transcriptome now of these epigenetic cancers, And so we know that there are some genes that become induced, and it's those genes that I told you that are in the signaling pathways that we know are triggered in these cancers and that trigger cell proliferation and loss of cell polarity. So what we are trying to set up to do is to, first inactivate polycomb and then measuring genome organization in these epicancers. And then we try to restore normal tissue by, restoring the expression of the downstream targets, at least the, one or two of the main downstream targets, and then see what happens to the architecture. And the other thing that we can do is, instead of making a null, like a just reduction of polycomb, we can start to express what we would call a structural mutants, which means proteins that, for instance, so these proteins that have IDR domains, internally disordered domains, intrinsic disordered domains, or another domain, which is a sum domain, which can induce the formation of aggregates. So we can just perturb these and see what happens. So it's like a good way to go. We have some work to do, but it's possible.
Terrific. All right, we've got a couple different questions here. So this is actually somewhat related, I guess, to what you were just talking about. So related to the first part of the talk, have you observed instances where the epigenome acted as a mutational buffer? Yeah. So PRC1-2 activity masking the oncogenic activity of specific mutations.
Yeah, this is also super interesting. And no, we haven't, we don't know actually. Among the genes that are dysregulated, we did find, although we did not find mutations in these in early stages, we did find overexpression of DNA replication, which might be expected, but also some DNA repair components. And so that's something which we are exploring. Do we see any increase in genomic stress? And then maybe, later on this could induce mutation. So it might rather be that you have an increased mutational burden at some stage of the cancer development. I have to say, what we can do with fly, although it's a limited system, we can still do xenografts, so like you do with a human. So you can propagate these tumors for many generations, and then we can look down the line what's the genome architecture going to be, and what's also the genome burden, mutational burden going to be.
Well, thanks. We are out of time. Do you have a few more minutes to take a couple more additional questions?
I would be happy to. Yeah, I would be happy to. Yeah, we can give it a go. Yeah.
Okay. One question here. Can PCG inactivation cause de-repression of target genes without altering CNDs or tags?
Oh, that's a brilliant one. Look, we have an initial answer. We are stunned because we have done micro-C in these cancers, and I tell you, we wipe out completely polycomb. Okay, when we do this depletion, we wipe out all the marks and we wipe out all the proteins. Okay, so it's a very clean situation, and what we see is all the long-range interactions are gone. Okay, or well, the overwhelming majority. But the short-range interactions within these domains are not very strongly perturbed, and that's very surprising. So you don't really just distend the whole chromatin. So yeah, it's although several of these genes start to express, that's something very curious, and we have to delve into that.
Thank you. A slightly different question: are modifications to epigenetic regulators heritable?
Modification, you mean that I don't know what they mean. Is it what post-translational modifications, or what do they mean?
I guess I was anticipating this or thinking about this in the context of heritable cancers, for example. Essentially, if there might be mutations that were passed down, essentially that would things like colorectal cancer, for example, or other types of heritable diseases.
Okay, so heritability. Well, now we haven't studied that because we induce it in a somatic tissue and that's it. But actually, that could be something to look into. And there's some interest whether the susceptibility, the cancer susceptibility changes. I mean, like these mutations, either it's too strong and you're dead if you pass it on the germline, or you have to be alive, the mutation must be really weak. Okay. But it is still something that people haven't looked, and it could be very interesting to look. If you have like partial loss of function mutation, do you have a susceptibility? And this is something absolutely great to look at. Yeah.
Thank you. Another question, in your description, the unmethylated CpG is being bound by a PRC1 complex, which triggers repressive marks. But how are active CpGs protected from this?
Yeah, absolutely. But this is the same. So that's also how, is it possible that polycomb is silencing? Yes, but sometimes it's not silencing. And so what we believe is that there are transcription factors that are doing the job, which are compatible with polycomb binding. And typically, whenever we see polycomb bound to active regions, it's always ataxic peak, so it's always open chromatin. And we did see motif enrichment for TFs, and in several cases it's pioneer factors. So what we believe is that factors that open, that are very proficient in opening up chromatin, even if there's nucleosomal structure, they are going to bind there, and then they are going to perturb the K27 trimethylation, but without perturbing the binding of polycomb. And then polycomb proteins can interact with each other, and that's how they can form loops. But now these loops cannot no longer be repressive, because these factors, they tend to come with histone acetylases, and so there might be competition with polycomb. That's how we envisage it.
Gotcha. Thanks. This question's a little bit, I was going to say, taking a different track, asking about long non-coding RNAs. So the question is, it's known that long non-coding RNAs can participate in 3D genome organization. Have you seen involvement of long non-coding RNAs and polycomb proteins in chromatin formations together?
We know that there are non-coding RNAs. It's true. So we met them in many of the polycomb target sites. We haven't explored this issue much more, but definitely possible. And as you know, it's a complicated field. You have activating and repressing non-coding RNAs. We have evidence, with some initial evidence, that several non-coding RNAs are expressed in the expressed genes. Actually, it's more like the sort of enhancer RNAs that would be associated, while other people in the mammalian field have published that non-coding RNAs can also help targeting of silencing polycomb components. And there we don't have much. The question is, we've looked at it in flies, and in this respect, are flies and mammals the same or not? That we don't know.
Gotcha. Thanks. Here's a question. Are loops anchored by activating polycomb loops different from repressing polycomb loops in terms of size, acetylation, dependence, or strength? Yeah,
so absolutely the marks, yes. But the size, not really. They are kind of pinpoint loops. Even when you have a domain, it's fully coated by K27 or ubiquitin H2A. But polycomb binds to specific regions, and it's there. It's the peak that is really forming the loop. So even repressive loops are kind of sharp. Now, people shouldn't confound this with the very long-range interactions that I've been talking about, because in this case, it's more like the whole domain. So when you have things happening over tens of megabases distance, typically like the whole repressive domain is getting together. But when you stay within one, actually I've shown you one example of this ring 1b bound regions in the HOX-D, which is in a way in the mammalian system is a bit more difficult because the polycomb peaks are not so nice. But still, you see that like the summit or the higher levels of signal, that's where you have the contacts between the HOX genes. So no, it's kind of sharp loops. in all cases.
We're gonna wrap up here in just a minute. Are polycomb H3K27 methylation 3 loops always associated with developmental or tissue specific genes?
Yeah, most of them. I mean, I'm like now I wouldn't put my hand on fire, but yeah, basically that's it. That's most of the genes that are bound are critical developmental genes that change in expression in different tissues in different stages of development. Yeah, I can't believe there's some housekeeping, but it's going to be an exception and not the rule. Yeah.
Here's a question about how do you define TAD insulation strength? Is it used the strength of the two sides of the boundaries, the corner peak, or is it the boundary frequency? Go ahead.
So there's various ways in which you can call TADs and TAD boundaries. We typically use this thing where we quantify. So we take one region, could be the boundary, and we quantify the contacts from the upstream to the downstream part, and the size of the upstream and downstream part that you take, you can vary it. And depending on the resolution of your map, the better the resolution, the smaller you can go. So for instance, you could take the site of interest and then look at the contacts that the 50 kb upstream make with the 50 kb downstream. If it's not a boundary, you're gonna have a lot of contacts. But if you stumble into a boundary, the contacts go down. And basically we plot the level of contacts you make with a sliding window, and whenever you have a trough, this is the boundary. And yeah, as I said, we can vary. Now with the good maps that we have, we can really go down to a few kbs. But yeah, if you have less reads, you have to open it up and maybe look at 100 and 100 kb left and right. So that's a quite efficient way to do it.
Thank you. I just had a question here in the I lost my question. Sorry.
That's quite a bit.
There are quite a bit of questions. So again, thank you for everybody for joining today. And if we're not able to answer your question, we'll reach out to you and follow up directly.
I'm happy to take some more, maybe by email or.
Okay. So this is a single cell question. Have you seen evidence of heterogeneity of TADs between individual cells using micro-C?
Okay. We haven't done single cell studies. So that's of course an interesting point. But as you know, single cell, high C, micro C, it's limited by the fact that you only, for a given point, you only have one contact that you can measure in the genome. So you really need to pile up cells to do that. So the best I think you can maybe do is doing lots of single cells, then try to cluster them somehow, and then to pile up the individual clusters to see. So kind of what you do with, RNA, but it's going to be much less effective than looking at RNAs, because still with RNAs you can get lots of reads for at least the highly expressed genes. In the case of micro-C, you're gonna have one contact or two per point. So, and when I say 1 or 2 per point, in fact, you're gonna see a few % of the possible contacts. So that is something that has not been exploited very much. We will see in the future if maybe the techniques improve. Definitely, there's a need to improve the quality of the libraries.
Yep. Well, thanks for that. All right. Well, that's it for today. We're out of time. There are several people that have asked questions. So once again, if you've asked a question, we will reply and reach out to you to continue that. If you would like to reach out directly to Dr. Cavalli, you're welcome to do so. And his contact information is on the screen, as well as our hosts, Dovetail Genomics. You can reach them at info@cantatabio.com. Once again, thank you all for joining today. And I wish you all the very best. Take care. Bye-bye.
Bye-bye.